we will be discussing the global control of an organism, which means that they express regulatory mechanisms that respond to environmental signals. An example of this would be the lac operon and seeing how cells respond when given more than one sugar. We will first discuss catabolite repression. Basically, bacteria will always use glucose first as a utilizable carbon source. The proteins of lac operon, including the enzyme beta-galactosidase, are required for using lactose. But the synthesis of these proteins is also subject to catabolite repression. As long as glucose is present, the lac operon is not expressed, and lactose is not used. However, when glucose is depleted, catabolite repression is abolished, and the lac operon expressed, and the cells grow on lactose.
Cyclic AMP is a key molecule in many metabolic control systems. Because it is derived from a nucleic acid precursor, it is a regulatory nucleotide. When glucose enters the cell, the cyclic AMP level is lowered, so a CRP protein cannot bind to DNA, and RNA polymerase fail to bind to the promoter region. In order for a lac gene to be transcribed, cyclic AMP level must be high so that the CRP protein will be able to bind to the promoter area of the lac operon.
Cyclic AMP is an important regulator molecule utilized in the GS and GI pathway. It is found and used by both eukaryotic and prokaryotic cells. The reaction of which cyclic AMP is synthesized is thus. Two phosphate groups on an ATP molecule is cleaved by adenylyl cyclase, and our second ring structure is formed on our final product. Glucose also plays an important role in the production of cyclic AMP. Because glucose is a good energy source, its precedence take precedence over cyclic AMP production. Basically, the cell will denote the energy used in ATP to cleave glucose rather than to make cyclic AMP. Glucose inhibits cyclic AMP synthesis by preventing CRP from binding to DNA and RNA. It also stimulates the transport of cyclic AMP out of the cell. Ultimately, as glucose concentration increases inside the cell, cyclic AMP concentration decreases.
Lac genes cannot be transcribed unless two requirements are met. I will be discussing those two requirements. As mentioned earlier in the video, cyclic adenosine monophosphate, CAMP, a second messenger, plays an important role. The level of CAMP must be high enough for the CRP protein, drawn here, to bind to the CRP binding site, drawn here in blue. This is a positive control because the regulatory protein or activator, which is CRP itself, activates the binding of RNA polymerase to DNA. This requirement is necessary since when glucose is present in the cell, CAMP level will not be as high, so CRP protein cannot bind. The presence of lactose tells you whether or not the repressor protein is bound. Hence, the second requirement is that the lactose or another suitable inducer must be present so that the lactose repressor, lac I protein, does not block transcription by binding to the operator. When lactose is there, it actually induces the repressor, drawn here in blue, to fall off. This is a negative control since lac I is responsible of repressing mRNA synthesis. If these two conditions are met, the cell is signaled that lactose is present, but there is no glucose. Under this circumstance, transcription of the lac operon begins.
I'll be discussing the overall regulation of the lac operon and how it utilizes catabolite repression. There are four main genes in the lac operon that encode for proteins. Lac I encodes a repressor. Lac Z encodes beta-galactosidase, which is responsible for breaking the bond in lactose between glucose and galactose. Lac Y encodes for lactose permease, which allows lactose to enter the cell. And Lac A encodes lactose acetylase, which transfers an acetyl group to lactose. The LAC-I protein is always translated. However, it can be active or inactive. When first translated, LAC-I is an active repressor, and as long as there is no inducer, such as in the top situation here, it will bind to the operator. Even if the CRP dimer with its cAMP and the RNA polymerase are bound to the DNA, LAC-I prevents transcription and translation of the LAC-Z, Y, and A genes. Thus, no proteins will be made and lactose cannot be broken down. If instead there is an inducer, such as in the bottom situation here, LAC-I becomes inactive as the inducer binds to it, and thus it unbinds from the operator. Now, if there is no CRP dimer and no RNA polymerase, nothing will happen, and the genes will not be will not go through transcription or translation, and lactose will still not be broken down. If the CRP dimer with its cAMP and RNA polymerase are present and bound to the DNA, only then can the LAC-Z, Y, and A genes be made into proteins, and then lactose can be broken down.